Topic of discussion is metabolism of carbohydrate. Carbohydrate are main part of our food. It is presented in the form of polysaccharide, disaccharide or monosaccharide. After digest, it will converted into simple form of sugar. It may be glucose, fructose or galactose. Main part is glucose. Glucose undergoes 4 types of metabolic activity. It will enter maybe glycolysis, glycogenesis, glycogenolysis, glyconeogenesis. What are the 4 metabolic activity? First one is glycolysis, second one is glycogenesis, third one is glycogenolysis, fourth one is glyconeogenesis. First process, glycolysis. What is glycolysis? Breakdown of glucose molecule to 2 molecules of pyruvate. This process takes place in cytosol of the cell. Cytosol is the aqueous component of cytoplasm. One glucose molecules will converted into 2 molecules of pyruvate. Then 2 ATP synthesized and reduced to NADH2. Now we will discuss about that metabolism of glycolysis. Glucose is converted into 2 molecules of pyruvic acid. First one is glucose. It will enter to phosphorylation process. ADP, adenosine triphosphate, is converted into adenosine diphosphate. So 1 molecules of inorganic phosphate will bind to the glucose molecule. So glucose 6 carbon component, then 6th position that phosphate molecule attached with the presence of magnesium ion and with the help of hexokinase enzyme. This reaction catalyzed by hexokinase enzyme. Second step, isomerization. What is isomerization? Nothing added, nothing removed. Glucose and fructose same molecular formula. So just glucose is converted into fructose 6-phosphate. Then again phosphorylation will occur. Adenosine triphosphate is converted into adenosine diphosphate. Then 1 molecules of phosphate will attach to the first position. So fructose 1,6 bisphosphate. Bis means 2 phosphate. First position and 6th position phosphate molecule is presented. This reaction catalyzed by phosphofructokinase enzyme. Then here cleavage breakdown process occur with the help of aldolase enzyme. This reaction catalyzed by aldolase. 3 carbon component will divided into 3 carbon component. Dihydroxyacetone phosphate, here glyceraldehyde 3-phosphate. So again this dihydroxyacetone phosphate with the help of phosphotriose isomerase enzyme, it will converted into glyceraldehyde 3-phosphate. So we get here 2 molecule of glyceraldehyde 3-phosphate. After that here up to end we get 2 molecules of all the component, 2 molecule of pyruvic acid. This glyceraldehyde 3-phosphate with the help of enzyme glyceraldehyde 3-phosphodehydrogenase, okay, it will converted into 1,3-bisphosphoglyceric acid. 1 molecules of phosphate attached here. Next process, the transfer of high energy phosphate group that was generated to ADP from ATP. This phosphorylation, that is addition of phosphate to ADP to give ATP, is termed as substrate level phosphorylation, as the phosphate donor is substrate 1,3-bisphosphoglyceric acid. Then 1,3-bisphosphoglyceric acid converted into 3-phosphoglyceric acid. Then this 3-phosphoglyceric acid with the help of phosphoglycerate mutase enzyme. Mutase, it will change place. Here third position, here it changed to second position. 2-phosphoglyceric acid. Then with the help of enolase enzyme. Here in addition of magnesium ion, enolase means one water molecule will remove here. It will converted into phosphoenol pyruvic acid. The product of this reaction, 2 molecules of 3-phosphoglyceric acid. This one is the first level phosphorylation. Then second reaction of glycolysis, where high energy product component is formed here. 2 molecules of phosphoenol pyruvate. Here enolization process will occur. After that finally we will get pyruvic acid. Finally we got this pyruvic acid catalyzed by pyruvate kinase enzyme. This first level substrate phosphorylation here, second level phosphorylation takes place. Now this pyruvic acid enter 2 type of reaction. Presence of oxygen means it will enter to mitochondria, undergo citric acid cycle. Absence of oxygen means pyruvate converted into lactate with the help of lactate dehydrogenase enzyme. This NADH2 converted into NAD+. This one again reutilized by glycolysis. This one is the notes format. So 1 molecule of glucose is converted into 2 molecules of pyruvic acid. Presence of oxygen means enter to citric acid. Absence means it will converted into lactate. Now the question is, what about galactose and fructose? Galactose is converted into glucose 1-phosphate. This glucose 1-phosphate is converted into glucose 6-phosphate. It will continue that cycle. Fructose is converted into fructose 1-phosphate. Fructose 1-phosphate again it will converted into 2 form, either glyceraldehyde dihydroxyacetone phosphate or glyceraldehyde. This dihydroxyacetone with the help of phosphotriose isomerase it converted into glyceraldehyde 3-phosphate. It will continue that cycle. Glyceraldehyde, here phosphorylation will occur. It will converted into glyceraldehyde 3-phosphate. It will continue this cycle. What is substrate level phosphorylation? Transfer of high energy phosphate group that was generated to ADP, adenosine diphosphate, from adenosine triphosphate. This phosphorylation, that is addition of phosphate to ADP to give ATP, is termed as substrate level phosphorylation, as the phosphate donor is 1,3-bisphosphoglyceride. This one is the substrate donor. Second level of substrate level phosphorylation, here donor is phosphoenol pyruvic acid. Glycolysis, step number 6 and step number 9, substrate level phosphorylation occur. Thanks for watching.